Right now, I mean, literally right this second inside your skull, you are running the most complex piece of hardware in the known universe.
Oh, absolutely. Nothing else even comes close.
Right. But here is the catch. You are building it using a blueprint that is constantly rewriting itself in real time while the architecture is actively being utilized.
Yes, which is quite hard to wrap your head around, really.
It's like imagine the electrical grid of a massive city. It's turning its own power plants on and off, changing the layout of the streets based on, who is driving on them, and just shutting down entire neighborhoods to redirect resources,
Yeah.
all while the city is fully populated.
It's a total biological paradox.
Right.
Because, we tend to conceptualize brain development like constructing a building. You lay the foundation, you erect the scaffolding, you wire the lights.
Right, step by step.
Exactly. But the central nervous system, it does not have the luxury of a closed construction site. It has to process information, regulate your physiology, and adapt to the environment while simultaneously building itself from the ground up.
And that is exactly what we are exploring on this deep dive, the functional genomics of human brain development.
It is a massive topic.
It is, but our mission today is to really pull back the curtain on the hidden molecular architecture that orchestrates this whole paradox. We are talking about the assembly of 86 billion neurons.
Wired together by hundreds of thousands of kilometers of myelinated axons.
Right, communicating across hundreds of trillions of synapses. And we are going to look strictly at the blueprint, the genomic and epigenomic regulatory networks that actually make all of this possible.
And before we get into the molecular weeds, it is really worth considering the massive evolutionary gamble that this construction project represents.
What do you mean by gamble?
Well, when you examine the human brain alongside other mammals, the most striking divergence is not merely its volume or even its structural complexity. It's the sheer protracted timeline of its development.
Right, it takes forever.
Exactly. We exhibit this uniquely extended gestational period, which is then followed by an incredibly long childhood and adolescence. Human brains literally take decades to reach full structural maturity.
I mean, you think about a horse, right? A foal is walking within hours of being born.
Spot on.
But a human infant is essentially helpless for years, relying entirely on external scaffolding from caregivers. That can't just be some evolutionary accident.
No, it is not. It's a fundamental evolutionary trade-off. This massive window of development, this protracted vulnerability, is precisely what allows the external environment to physically shape our cognitive, emotional, and social capacities.
So the brain remains intensely plastic just so it can be sculpted by our experiences.
Precisely. But that long, highly intricate window makes the human brain exceptionally fragile. It demands the flawless regulation of millions of molecular events over decades.
And if those events fall out of sync?
Then the system becomes highly susceptible to psychiatric and neurodevelopmental conditions. If the timing is off by even a fraction, the architecture changes.
Okay, let's unpack this. To understand how that system can falter, we really need to map the sheer scale of the instruction manual we are dealing with.
Right, the genome.
We were looking at the human nuclear genome, which consists of, what, roughly 6.2 to 6.4 billion base pairs?
Yes. And this is actually where classical genetics threw us quite a curveball. For decades, the assumption was always that the complexity of an organism would just linearly correlate with its number of protein-coding genes.
More complex animal, more genes?
Exactly. It makes intuitive sense. But out of those billions of base pairs, the human genome only contains roughly 19,982 protein-coding genes.
Which is, that is roughly the same number as C. elegans, right? That microscopic nematode worm.
Yes.
So how does that mathematical reality even make sense? How does that limited inventory build a human cortex?
Well, the complexity does not arise from the number of raw materials. It comes from how those materials are spliced, edited, and deployed. First, we have alternative splicing. Over 90% of human protein-coding genes possess splice variants.
So they can be mixed and matched?
Yes. The transcriptional machinery can just cut and paste the exons of a single gene in multiple configurations to generate entirely distinct protein isoforms. And crucially, the human central nervous system exhibits the highest splicing complexity of any organ in the body.
So the genome isn't a static dictionary. It's highly combinatorial.
Highly. And furthermore, those protein-coding regions represent only a tiny fraction of the genome. We also have over 26,000 non-coding RNA genes.
So these don't make proteins at all.
No, these transcripts do not translate into proteins, but they serve as critical regulatory molecules, microRNAs, and long non-coding RNAs that modulate the stability and translation of other transcripts.
Right.
But the true engines of complexity, the real directors of this whole process, are the cis-regulatory elements, or CREs.
You mean the promoters and the enhancers?
Exactly. Current genomic annotations suggest the human genome harbors roughly 70,000 promoters. Those are the proximal sequences required to initiate transcription, and a staggering 400,000 putative enhancers.
400,000.
Yes. And enhancers are these distal DNA sequences that act as the master switches. They dictate the precise spatial and temporal coordinates of gene expression. They determine when and where a gene is activated.
Right. So to actually read this active blueprint, clinical neuroscience has had to develop an entirely new methodology. We aren't just looking at static DNA sequences anymore. We need to capture the genome in motion.
Which is exactly why the shift from structural genomics to functional genomics relied entirely on the advent of high-throughput sequencing technologies.
Yeah.
We moved from just looking at candidate genes to interrogating the entire transcriptome and epigenome simultaneously.
And we should break down this alphabet soup of technologies for a second because it's fascinating. If we think of the genome as a sprawling, highly complex theatrical script, RNA-seq, or RNA sequencing, is essentially our way of documenting exactly which lines are being spoken by the actors on stage at any given millisecond.
That is a brilliant analogy.
Right.
Yes, RNA-seq quantifies the active transcripts, both coding and non-coding present, in a tissue sample, or nowadays, even within a single cell. It provides an unbiased, quantitative snapshot of the cellular state.
But RNA-seq only tells us the output, right? The spoken lines. To understand the input, the actual regulation, we need ATAC-seq, assay for transposase-accessible chromatin.
Right.
So following the theater analogy, if RNA-seq is the spoken dialogue, ATAC-seq tells us which stages have the curtains physically pulled back. It uses a hyperactive transposase enzyme to tag the open chromatin.
Yes, the regions of DNA that are physically unwound from their histone spools and exposed to the transcriptional machinery. And that open chromatin maps almost perfectly to the active promoters and enhancers we just discussed.
But even knowing which curtains are open isn't quite enough, is it? We need to know who is standing on the stage.
Exactly. We need to know which specific regulatory proteins are binding to those regions. That is where PSYCH comes in. Chromatin immunoprecipitation sequencing.
Right, that's where we locate the directors and the stage managers. PSYCH uses specific antibodies to pull down DNA that is physically bound to a target protein, like a specific transcription factor or a modified histone.
It allows us to map the precise genomic coordinates where a regulatory protein is exerting its influence. And finally, we have the repressive marks. We use DNA methylation arrays or whole genome bisulfite sequencing, WGBS.
The silencers?
Yes. This maps the epigenetic modifications, specifically the addition of methyl groups to the DNA that generally condense the chromatin and silence gene expression.
Okay, so we have this incredible high-throughput toolbox that captures the open chromatin, the transcription factor binding sites, the repressive methylation, and the final RNA output. When researchers actually deploy these tools to map the developing human brain, what does the active landscape look like? We are essentially talking about a four-dimensional map here, 3 spatial dimensions plus time.
And the temporal data fundamentally rewrites how we view neural development. When you look at the entire transcriptome across the human lifespan, from early embryogenesis all the way to late adulthood, you find that at least 86% of all protein-coding genes are deployed in the central nervous system at some point.
Wait, 86%?
Yes.
That implies the brain relies on almost the entire genetic repertoire of the human species just to assemble itself.
It does. It is a staggering metabolic and regulatory demand. And out of that 86%, roughly 90% are differentially expressed. Meaning their expression levels are not static. They fluctuate massively, depending on the specific brain region or the precise developmental stage.
So the brain is absolutely not simply activating a set of, neural genes and just leaving them switched on.
Far from it. And the temporal dynamics are incredibly asymmetrical. The vast majority of transcriptomic changes occur during 2 critical, highly vulnerable windows.
Okay, what are those?
The first is the early fetal transition, spanning post-conception weeks 6 to 10. And the second is the massive transition between late fetal development and infancy.
So we see explosive molecular shifts just before birth and right around the time the infant actually enters the world. How does that compare to the rest of the lifespan? Like, what happens when we are adults?
It's remarkably quiet. If you track gene expression changes across the decades of human adulthood, the variance is almost flat. Less than 1% of genes significantly alter their expression profile over 40 years of middle adulthood.
Wow.
Yeah, the heavy lifting, the immense molecular choreography, it is heavily front-loaded in our developmental timeline.
So if the temporal shifts are that drastic, what about the spatial landscape? If we look at the multidimensional scaling data, which for you listening essentially clusters these tissue samples based on how similar their transcriptomes are, do different brain regions look genetically distinct from the very start?
The spatial divergence is profound, but it is not uniform. The cerebellum, for instance, immediately isolates itself in the data. It is the most transcriptionally distinct structure in both the developing and the adult brain.
Which makes sense given its unique cellular architecture, right? All those dense granule cells.
Exactly. The thalamus and striatum follow as distinct clusters as well.
But clinically, the focus usually centers on the neocortex. The seat of executive function, associative learning, all are complex cognition. How does the prefrontal cortex construct itself over time?
If you trace the functional ontology of the prefrontal cortex, meaning categorizing the active genes by their biological roles, you observe a tightly orchestrated relay race. During the embryonic phase, the transcriptome is heavily dominated by genes driving cell proliferation.
So the radioglia and neural progenitor cells are multiplying rapidly.
Right, generating the raw cellular inventory.
But then those cells have to actually become specific types of neurons.
And right on cue, as we transition into the mid-fetal period, those proliferation networks are aggressively downregulated, and the genes driving neuronal differentiation and migration spike.
So the cells are migrating along the radial glial fibers and taking on their specific layer identities.
Precisely. And then later, during the perinatal period and extending into the first year of life, a completely different transcriptional module emerges, myelination.
The oligodendrocyte lineage kicks in to insulate the axons.
Correct. The myelination network rises sharply perinatally and plateaus around age 1, which reflects the physical maturation of the white matter tracks necessary for rapid signal conduction.
Okay, so we have this beautiful temporal relay race, but if we compare different areas of the neocortex, say, comparing the prefrontal cortex to the primary visual cortex in the occipital lobe, how different are those two regions at a molecular level as they develop?
Well, this reveals one of the most conceptually elegant models in all of neurodevelopment. It's called the neocortical temporal hourglass.
An hourglass.
Yes. If you quantify the interareal differences, the actual transcriptomic distance between distinct cortical regions, you find massive divergence during mid-fetal development.
Why mid-fetal?
Because that is the critical window for regional specification. The nascent circuits are being assembled, and the prefrontal cortex must establish a radically different cellular architecture and connectivity profile compared to, say, the sensory cortices. The regions are actively differentiating themselves.
So it's wide at the top of the hourglass, but then it pinches inward.
Yes. As development progresses into the late fetal and early childhood stages, those massive regional differences suddenly collapse. The distinct areas of the cortex become transcriptionally much more homogeneous.
Which seems totally counterintuitive. Why would a toddler's prefrontal and visual cortices look more genetically similar than they did when they were in the womb?
Because the biological priorities shift from regional specification to global pancortical processes. During early childhood, the entire cortex is undergoing massive synaptogenesis, dendritic arborization, and the maturation of allocyte networks. These are fundamental biological requirements for every cortical region. So the shared molecular demands of building all those synapses completely overshadow the subtle regional differences, pulling the transcriptomic profiles together.
So the entire system is essentially running the exact same wiring subroutines. But as the child enters adolescence, does the hourglass open up again at the bottom?
It does. During adolescence and early adulthood, the interareal differences widen once more. This divergence likely reflects the final, specialized maturation of associative cortices compared to primary sensory areas. It's fine-tuning the higher-order cognitive and socio-behavioral circuits.
I want to push back on a mechanistic detail here, though. You mentioned the prefrontal cortex differentiating itself during that mid-fetal window. How does a migrating neural progenitor physically know it is situated in the anterior portion of the brain? I mean, it doesn't have a GPS.
No, but it reads the local environment, relies on morphogen gradients. These are diffusible signaling molecules that establish concentration maps across the developing tissue. A classic example detailed in the functional genomic data is retinoic acid signaling. Transcriptomic mapping of the human midfetal cortex revealed a stark rostrocaudal gradient of retinoic acid synthesis.
Rostrocaudal, meaning front to back.
Yes, the molecular machinery to produce retinoic acid is highly enriched in the anterior prefrontal cortex, and the concentration steadily tapers off as you move posterior.
So a cell literally reads the ambient concentration of retinoic acid to determine its spatial coordinates. Like, if the concentration is high, I must be in the frontal lobe, and I should activate prefrontal specific transcription factors.
That is exactly the mechanism, and we can actually validate this functionally. When researchers selectively ablate the enzymes responsible for this retinoic acid gradient in embryonic mice, the anterior neural tissue fails to specify properly.
Real?
Yes, the prefrontal cortex loses its unique identity entirely, and the highly specific corticostriatal and thalamocortical connectivity networks are fundamentally degraded.
That front-to-back chemical map makes perfect sense. But here is where it gets really interesting. What about lateralization? Because clinically, we know the human brain is highly asymmetrical in its function.
Of course.
Language dominance is typically lateralized to the left hemisphere, while spatial attention and prosody are heavily right lateralized. One would expect the left and right hemispheres to possess radically different baseline transcriptomes to account for these massive functional differences.
It is a totally logical assumption, but the high-throughput data directly contradicts it. Despite the profound functional and anatomical lateralization in the mature human brain, population-level transcriptomic analyses reveal that the left and right hemispheres are remarkably symmetric from early fetal development straight through to adulthood.
Wait, there are no major left hemisphere-specific gene expression networks?
None that are robustly detectable at the whole tissue level. I mean, there may be hyperlocalized transient asymmetries in specific cellular subpopulations that our current assays miss, but at a macro level, the blueprint builds a highly symmetric foundation.
Which carries a fascinating clinical implication. If the baseline genetic blueprint is symmetric, then the functional lateralization we observe in language or motor dominance must be driven heavily by non-transcriptional mechanisms.
Exactly.
Yeah.
It strongly implicates experience-dependent plasticity. The neural activity itself, the asymmetric sensory inputs, and the physical firing of the circuits likely drives the functional specialization of the hemispheres over time. The genome provides the scaffolding, but the physiological activity dictates the final architecture.
Let's examine the actual mechanics of that scaffolding, though. We have established that enhancers and promoters dictate this 4D temporal map, but how does an enhancer actually execute that control?
To understand gene regulation, we have to discard the linear view of DNA and think in three-dimensional physics.
Okay.
A core promoter is a DNA sequence situated immediately adjacent to the transcription start site of a gene. It is the landing pad for RNA polymerase. However, the regulatory elements, the enhancers that actually dictate the tissue specificity and timing, can be located hundreds of thousands of base pairs away, sometimes even on completely different chromosomes.
So how does an enhancer physically communicate with a promoter if they are separated by a massive expanse of genomic distance?
Through dynamic three-dimensional chromatin looping. An active enhancer is bound by specific transcription factors. These factors, alongside architectural proteins like the cohesin complex and CTCF, physically fold the intervening DNA. They grab the distal enhancer and lure it through three-dimensional space until it makes physical contact with the core promoter.
So it is a mechanical tethering. The DNA literally bends back on itself.
Yes, and this creates a highly competitive, sterically constrained environment. The source text actually provides a brilliant mechanistic case study regarding the regulation of the FESF2 gene.
What is the clinical relevance of FESF2?
FESF2 is a master regulatory transcription factor required for the specification of subcerebral projection neurons. In the human cortex, these are the massive upper motor neurons located in cortical layer 5.
Right.
Their axons form the corticospinal tract, descending all the way down to the spinal cord to govern voluntary motor control. If a developing brain fails to express FESF2, the corticospinal tract simply does not form.
So the developmental imperative is absolute. FESF2 must be turned on in layer 5. But equally important, it must remain OAF in layer 6 and the apocortical layers. Otherwise, the wiring topology would be complete chaos.
The precision required is astonishing. To achieve this, the genome utilizes an evolutionarily conserved distal enhancer known as E4. The E4 enhancer is the focal point of an intense molecular tug-of-war.
Who is pulling the rope?
During development, two transcription factors, SOX4 and SOX11, act as potent activators. They bind to the E4 enhancer, initiate that 3D chromatin loop, and drive FESF2 expression.
But if SOX4 and SOX11 are present throughout the deep cortical layers, what stops them from turning FESF2 on in the wrong place?
A third transcription factor, SOX5. SOX5 acts as a targeted repressor. Crucially, SOX5 has an affinity for the exact same binding motif on the E4 enhancer as the activators. It physically competes with SOX4 and SOX11 for occupancy on the DNA.
Oh, I see. So if SOX5 binds first, it sterically blocks the activators, completely preventing the chromatin loop from forming.
Precisely. It is competitive inhibition at the genomic level. But the fail-safes are even more robust than that. What about layer 6?
Right, the neurons in layer 6 project to the thalamus, not the spinal cord. They absolutely must not express FESF2.
Exactly. In layer 6, a different transcription factor called TBR1 is highly expressed. TBR1 binds to a completely separate regulatory element downstream of FESF2 and acts as a dominant silencer.
So even if the SOX activators somehow manage to bind to the E4 enhancer, TBR1 effectively cuts the power to the promoter.
It creates a perfectly locked regulatory grid. The interplay of competitive activators and repressors at the E4 enhancer, combined with dominant silencers in adjacent layers, flawlessly restricts FESDEF2 expression exclusively to the layer 5 motor neurons.
Which brings us to perhaps the most compelling concept in functional genomics, evolutionary divergence. If the basic mammalian blueprint is largely conserved, how did the human prefrontal cortex achieve such profound synaptic complexity?
Well, the CBLN2 gene provides a phenomenal model for this.
CBLN2, the human superpower.
Quite literally. CBLN2 encodes a protein known as a synaptic organizer. It is secreted to the synaptic cleft and physically bridges the presynaptic and postsynaptic membranes, stabilizing the connection. Now, in a standard mammalian model, like the mouse, CBLN2 expression is strictly confined to the upper layers of the prefrontal cortex.
It is entirely absent from the deep cortical layers in rodents.
Yes. But when transcriptomic profiling was performed on the human mid-fetal prefrontal cortex, the data revealed a massive spatial expansion. In humans, CBLN2 is highly expressed across all layers of the cortical plate, including those deep projection layers.
How does a species acquire that kind of widespread transcriptional expansion? Did we evolve an entirely new promoter?
We didn't gain genetic material, we lost it. Comparative genomics identified a hominy-specific deletion, a tiny sequence of DNA missing in humans, chimpanzees, and bonobos, but present in other mammals. This deletion occurred directly within the primary enhancer that regulates CBLN2.
But how does deleting a segment of an enhancer result in an increase in gene expression?
It depends entirely on what was deleted. The missing segment of DNA contained the specific binding motifs for the SOX5 repressor.
The exact same SOX5 repressor that shuts down FESF2.
The very same. In the ancestral mammalian genome, SOX5 binds to the CBLN2 enhancer in the deep cortical layers and represses the gene. During hominid evolution, a random mutation deleted that specific binding site. Without the repressor's landing pad, SOX5 can no longer bind.
The genomic break line was severed.
Exactly. The gene was structurally de-repressed. As a result, CBLN2 expression expanded into the deep layers of the prefrontal cortex, and the morphological consequences are striking. Because of this widespread synaptic organizer, human prefrontal pyramidal neurons develop a disproportionately higher density of dendritic spines compared to other primates or rodents.
That's wild. We achieved a massive increase in synaptic connectivity and computational power, not by inventing a new gene, but by breaking a regulatory stop sign.
And this mechanism was elegantly validated in vivo. Researchers engineered a transgenic mouse line where the native mouse CBLN2 enhancer was replaced with the human version, the version containing the hominy-specific deletion.
Now what happened?
Those mice developed human-like pancortical expression of the gene, resulting in a stable, quantifiable increase in dendritic spine density in their prefrontal cortex.
That perfectly illustrates how functional neuroanatomy is dictated by regulatory architecture. But the static DNA sequence, even with its promoters and enhancers, is only the foundational layer. We have to address how the environment physically interacts with this blueprint through epigenetic modifications.
Right. Epigenetics bridges the gap between the static genome and the dynamic environment. These are covalent chemical modifications to the DNA molecule or the histone proteins that alter chromatin accessibility without actually changing the underlying nucleic acid sequence.
And the most heavily researched of these modifications is DNA methylation.
Yes, specifically the addition of a methyl group to the cytosine base. In classical mammalian epigenetics, this methylation almost exclusively occurs at CpG-10 nucleotides, where a cytosine is immediately followed by a guanine.
And CpG methylation generally acts as a repressive mark, right? It condenses the chromatin, physically blocking transcription factors from binding?
He does. And we see massive reconfigurations of the CpG methylation landscape during the transition from fetal to postnatal life, heavily correlating with the shifting cellular composition of the developing brain. But the truly revolutionary finding in human neurodevelopment involves a different kind of methylation, non-CpG methylation, referred to as MCH.
MCH. What distinguishes MCH from standard CpG methylation?
Two critical factors. First, MCH exhibits profound cell-type specificity. It accumulates abundantly in the genomes of neurons, but it is virtually absent in glial cells.
Interesting. And the second?
The temporal kinetics are entirely different. Standard CpG methylation is largely established during early embryogenesis. MCH, however, accumulates specifically during the early postnatal developmental window.
The early postnatal window. That is the peak phase of synaptogenesis and experience-dependent pruning. The infant is processing sensory input, forming attachments, and establishing baseline neural circuits.
And that is precisely why MCH is so significant. It is actively laid down in mature postmitotic neurons while they're forming networks. The data suggests that MCH accumulation contributes to the human-specific downregulation of certain gene networks during this critical period.
So early postnatal neural activity driven by environmental experience may directly influence how these repressive MCH marks are distributed across the neuronal genome.
Yes, it provides a direct biochemical mechanism for how early life adversity or enriched environments could permanently alter the physical structure of the brain.
Yeah.
The experience is essentially codified into the epigenome.
It is the biological physicalization of memory and environment, but epigenetic regulation is not a one-way street, is it? If the system can lay down repressive marks, it must have a mechanism to remove them. Active DNA demethylation must be just as crucial for neurodevelopment. But how does a neuron actively remove a methyl group? That is a highly stable covalent bond.
It requires a specialized family of enzymes known as the TET proteins, 1011 translocation methylcytosine dioxygenases. The TET enzymes do not simply rip the methyl group off, they sequentially oxidize the repressive 5-methylcytosine mark, converting it first into 5-hydroxymethylcytosine, or 5-HMLC.
So 5-HMLC is an intermediate state. It's the epigenetic mark being primed for removal.
Yes. And surprisingly, 5-HMLC is remarkably stable in the brain. It accumulates heavily in mammalian neurons shortly after birth, specifically when the cells transition from active division to terminal differentiation.
What are the clinical consequences if the TET enzymes fail to convert 5-HMLC to 5-HMLC?
The developmental trajectory simply halts. Animal models have demonstrated that if mature neurons are unable to continuously generate 5-HMLC following the first week of postnatal life, they fail to achieve terminal maturation. The repressive marks remain locked in place, the necessary adult gene networks cannot be accessed, and the neuron remains functionally immature.
If the epigenetic scaffolding cannot be dismantled, the mature circuits never come online, which serves as a perfect conceptual bridge to the clinical reality of psychiatry and neurodevelopment. Because when you map a system this complex reliant on spatial morphogens, competing transcription factors, evolutionary enhancer deletions, and dynamic epigenetic editing, the sheer number of failure points is staggering.
And this is where functional genomics directly informs clinical reasoning. It reframes our entire understanding of etiology. Psychiatric and neurodevelopmental disorders are rarely the result of a single broken part. They represent a perturbation in the spatial and temporal assembly of the system.
And the timing of the clinical presentation often reflects when that perturbation occurred. Neurodevelopmental conditions like autism spectrum disorder or severe intellectual disabilities typically manifest very early in childhood.
Because they frequently stem from disruptions in the foundational developmental processes, neurogenesis, early cell migration, or the initial waves of cortical synaptogenesis. The architectural foundation is fundamentally altered.
Conversely, conditions like schizophrenia or bipolar disorder typically do not present clinically until late adolescence or early adulthood,
Which points toward dysregulation in the later stages of maturation. These conditions often implicate disruptions in late-stage synaptic pruning, the final waves of prefrontal myelination, or the delayed divergence of the associative cortices we saw in the neocortical hourglass model.
Yeah.
The basic structure is intact, but the final high-order network refinement fails.
Let's examine the specific mechanisms of these failures, starting with a monogenic condition, Fragile X syndrome. This is the leading inherited cause of intellectual disability and a massive genetic risk factor for autism.
Right. Fragile X is caused by the silencing of a single gene on the X chromosome, FMR1. The FMR1 gene encodes a protein called FMRP. Now, FMRP is not a structural component of the synapse. It is an RNA-binding protein.
Okay, so what does it do?
Its primary biological function is to bind to other messenger RNAs and regulate their transport to the synapse and their subsequent translation into proteins.
It's a master translational regulator, so losing FMRP does not just break one circuit, it disrupts the translation of hundreds of downstream targets.
Exactly. And functional genomics allowed us to identify those specific targets in the human brain. One of the most critical targets is the mRNA transcript for NOS1 neuronal nitric oxide synthase 1.
What role does NOS1 play in a developing human cortex?
During mid-fetal development, NOS1 protein is expressed in a highly specific anatomical pattern. It is restricted to the layer 5 subcerebrally projecting pyramidal neurons, the same motor output neurons we discussed earlier.
Right.
Furthermore, it is arranged in alternating mini-columns, specifically within cortical regions associated with speech, language, and complex social cognition.
So it is highly concentrated in the exact circuits that are functionally impaired in Fragile X in autism.
Yes. During neurotypical development, FMRP actively binds to the NOS1 mRNA and promotes its translation into protein during the critical window of synaptogenesis.
Oh.
But in a patient with Fragile X, FMRP is absent.
Without the translational activator, what happens to the NOS1 protein level?
They collapse. The mRNA is present, but it is not efficiently translated. Consequently, the NS1 protein levels are dramatically reduced in those specific layer 5 mini-columns. The biochemical profile of the language and social circuits is fundamentally degraded. It perfectly links a single gene mutation to a hyperspecific functional neuroanatomical deficit.
That is a brilliant example of a single broken node. But clinical psychiatry more frequently deals with polygenic complexity. Let's look at Down syndrome, trisomy 21. An entire extra copy of a chromosome. One would assume the primary pathology lies in the neurons themselves.
That was the prevailing hypothesis for decades, yes. Trisomy 21 causes massive genome-wide transcriptional dysregulation due to gene dosage effects. But when researchers applied unbiased transcriptomic network analysis to postmortem Down syndrome brain tissue, the primary cellular target was unexpected.
It wasn't the neurons.
No. The most profound dysregulation was not in the mature neurons, but within the oligodendrocyte lineage.
The glial cells responsible for myelination.
Yes. The single-cell RNA sequencing data revealed a stark maturation arrest. The transcriptional networks associated with immature oligodendrocyte progenitor cells were abnormally upregulated, while the networks required for mature myelinating oligodendrocytes were severely suppressed.
So the cells are proliferating, but they are failing to differentiate. They never mature enough to wrap the axons in myelin.
Exactly. And this transcriptomic finding prompted neuropathologists to re-examine the physical tissue. They confirmed prominent hypomyelination and severe disorganization of the white matter tracts, particularly in the prefrontal cortex.
And without that insulation.
Right, this lack of insulation heavily impairs rapid action potential conduction, which likely serves as a primary driver of the cognitive processing delays seen in Down syndrome.
It completely shifts the therapeutic target from the neuronal synapse to the white matter infrastructure. And this approach of mapping genes to spatial and temporal coordinates yielded an even more profound paradigm shift for idiopathic psychiatric conditions.
You were referring to the convergence of GOO's data with the spatiotemporal transcriptomic atlases.
Yes. Genome-wide association studies have identified hundreds distinct low-penetrance risk loci for autism spectrum disorder and schizophrenia. For a long time, it looked like random static across the genome. How do you build a clinical model when a disorder is linked to 200 different genes doing completely different things?
You look for the biological bottleneck. Researchers took those hundreds of GOO's risk genes for ASD and mapped them onto the 4D transcriptome. They asked, at what point in development and in what specific cell type are all of these seemingly unrelated risk genes actively co-expressed?
And the data isolated a specific nexus.
It did. The risk networks for autism spectrum disorder heavily converged on deep-layer cortical projection neurons within the frontal lobe, specifically during the mid-fetal developmental window.
That exact same spatial and temporal coordinate we have been coming back to. What about schizophrenia? It presents decades later. Surely its genetic risk converges somewhere in late adolescence.
That was the expectation. But when they mapped the schizophrenia GWAS data, it converged on the exact same developmental node, mid-fetal, deep-layer prefrontal projection neurons.
So the architectural vulnerabilities for both autism and schizophrenia, two radically different clinical presentations, are rooted in the exact moment those early cortical circuits are first reaching out to form connections in the womb.
It suggests a shared foundational vulnerability. If that specific midfetal choreography is perturbed by a high burden of risk alleles, the network develops a latent fragility. In autism, the developmental deviation is often severe enough to manifest early.
But in schizophrenia,
In schizophrenia, the fragile network may compensate through childhood, but catastrophically fails during the intense synaptic pruning and myelination demands of late adolescence.
it fundamentally reframes schizophrenia from a degenerative adult disease to a delayed onset neurodevelopmental disorder.
Absolutely. And we must also consider the mutations that do not fall within the coding genes at all. The non-coding regulatory elements we discussed earlier are equally critical failure points.
Right, a mutation in an enhancer or promoter.
A prime clinical example is perisylvian polymicrogyria. This is a severe congenital malformation of the cortex. Polymicrogyria translates to many small, abnormally formed gyri, or folds, on the surface of the brain.
In the perisylvian region.
That encompasses the critical language networks like Broca's and Wernicke's areas. Patients suffer from severe language impairment and intractable epilepsy.
What actually drives the malformation?
It is linked to a gene called GPR56, which is essential for the proliferation of neural stem cells. However, in these specific patients, the GPR56 coding sequence is perfectly intact. The mutation is a microscopic 15-base pair deletion within a specific enhancer located entirely upstream of the gene.
A 15-base pair deletion in a genome of 6 billion.
Yes, and that specific enhancer is the one responsible for driving GPR56 expression exclusively in the perisylvian cortex during embryonic neurogenesis. Because of that tiny deletion, the required transcription factors cannot bind.
So the local switch is broken?
Exactly. The stem cells in that specific region do not proliferate adequately, the radioglial scaffold collapses, and the cortex folds into a chaotic, microgyric pattern.
It is the ultimate demonstration of modular genetic control. The rest of the brain forms normally because the other enhancers for GPR56 are fine.
Which brings us to the final and perhaps most clinically actionable layer of dysregulation: epigenetic pathology in psychiatry. If the genome is the hardware, the epigenome is the software, and in conditions like schizophrenia, the software is deeply corrupted.
How does that epigenetic corruption manifest in the post-mortem schizophrenic brain?
We consistently observe targeted hypermethylation and excess of those repressive marks on the promoters of critical neurodevelopmental genes. One well-documented target is RELN, the gene encoding relin.
Relin is an extracellular matrix protein, right?
Yes, absolutely vital for neuronal migration during development and for synaptic plasticity in the adult cortex.
If the RALN promoter is hypermethylated, the gene is functionally silenced. The neurons lack the plasticity required for normal cognitive function.
Precisely. Another highly replicated finding involves the GAD1 gene. GAD1 encodes the enzyme responsible for synthesizing GABA, the brain's primary inhibitory neurotransmitter.
The brakes of the central nervous system.
Yes. In the prefrontal cortex of patients with schizophrenia, the GAD1 promoter is heavily hypermethylated, and the associated histones carry repressive modifications. Consequently, GABA synthesis plummets.
If you lose the inhibitory tone, the excitatory pyramidal neurons become hyperactive. The signal-to-noise ratio in the prefrontal cortex degrades, leading directly to the working memory deficits and disorganized thought processes characteristic of the disorder.
And we are beginning to understand why this epigenetic machinery fails in the first place. Genetic sequencing has identified severe, albeit rare, loss-of-function mutations in a gene called SCDD1A in schizophrenic cohorts.
What is SCD1A?
It is a chromatin modifier. It is the actual enzyme complex responsible for methylating specific histone tails to regulate gene expression. It is a core component of the epigenetic toolkit.
So a mutation in SCD1A means the epigenetic editing machinery itself is fundamentally broken.
Exactly. Mice engineered with SETD1A mutations exhibit profound deficits in working memory and severely degraded cortical connectivity. When the overarching epigenetic regulators fail, the entire transcriptomic landscape of the prefrontal cortex descends into absolute chaos.
This entire exploration requires us to step back and view psychiatric and neurodevelopmental conditions through a much more sophisticated lens. We have to move past the simplistic model of chemical imbalances that can be corrected by simply topping off serotonin or dopamine.
Yes, definitely.
We are looking at architectural, temporal, and molecular divergences that span from the first weeks of gestation to the final stages of cortical maturation in our 20s.
That is the core clinical paradigm shift. Localization is critical, knowing which layer of the cortex or which specific circuit is affected. But connectivity and temporal regulation explain the true complexity. A psychiatrist is not just treating a symptom, they're interacting with the downstream consequences of a highly regulated, interconnected genomic network that has been dynamically shaped by the patient's environment over a lifetime.
It is a profound framework for clinical reasoning. And before we conclude this deep dive, I want to introduce a final concept from the source material that entirely destabilizes how we view the biology of the self: somatic mosaicism.
Oh, this is a rapidly expanding frontier in functional genomics.
The fundamental assumption of human biology is that we possess a single monolithic genome. The DNA in a skin cell is identical to the DNA in a hepatic cell, which is identical to the DNA in every neuron.
That is the classical Mendelian assumption, yes.
But the high-throughput sequencing of single neurons is proving that assumption false. Every time a cell divides, the DNA replication machinery must copy 6 billion base pairs. It is highly accurate, but it is not perfect. Errors occur, de novo somatic mutations.
And because the neocortex is generated through billions of rapid cell divisions from a small pool of embryonic progenitor cells, any mutation that occurs in an early radioglial cell will be permanently passed down to every single neuron it produces.
It creates a clonal lineage of genetically distinct cells, which means your brain is not genetically uniform, it is a mosaic.
Exactly. The neurons in your left prefrontal cortex may carry a distinct set of somatic mutations that are entirely absent from your right visual cortex.
The implications for psychiatry are staggering. A patient could suffer from a severe neurodevelopmental disorder driven by a catastrophic mutation that only exists in a 15% patch of their frontal lobe. If you sequence their blood, their DNA will look perfectly normal.
The clinical phenotype is driven entirely by a hidden, highly localized genetic mosaic within the cranium. We are only just developing the single-cell technology sensitive enough to detect these deeply hidden variations.
It forces us to ask profound questions about human individuality. How many of our unique cognitive quirks, our specific talents, or our latent psychiatric vulnerabilities are the direct result of microscopic genetic mosaics hidden deep within the folds of the neocortex? We are not a single blueprint. We are a patchwork of trillions of microscopic variations.
It reinforces the reality that the brain is an evolving ecosystem, not a static machine.
Absolutely brilliant. Well, before we sign off, I want to leave you, the listener, with a few single best answer questions to test your clinical reasoning on these concepts. Think about what we've discussed today. I'll read the questions, give you a brief pause, and if you need to, jump back into the chapter to verify your thinking. Ready? Question 1. A patient with a severe neurodevelopmental disorder is found to have a genetic mutation that disrupts the function of the cohesin complex. Based on the principles of functional genomics, which of the following molecular processes is most likely to be directly impaired? Is it A, the splicing of pre-mRNA transcripts, B, the oxidation of 5-methylcytosine to 5-hydroxymethylcytosine, C, the three-dimensional looping of distal enhancers to core promoters, or D, the reading of the rostrocaudal retinoic acid gradient?
Think about what cohesin physically does to the DNA strand.
Right. Question 2. A post-mortem transcriptomic analysis of a patient with Down syndrome reveals massive dysregulation in the prefrontal cortex. According to single-cell data, which specific cell lineage exhibits the most profound maturation arrest driving the observed hypomyelination? Is it A, radial glial cells, B, deep layer pyramidal neurons, C, oligodendrocyte progenitor cells, or D, cerebellar granule cells?
This one fundamentally changes the therapeutic target for trisomy 21.
Question 3. Genome-wide association studies, or GWS, for both autism spectrum disorder and schizophrenia show a convergence of risk alleles. In which spatiotemporal node do these risk networks predominantly overlap? A, cerebellar Purkinje cells during early infancy, B, deep layer cortical projection neurons during mid-fetal development, C, prefrontal astrocyte networks during late adolescence, or D, upper layer cortical interneurons during the perinatal transition?
Remember the bottleneck concept we discussed?
Question 4. The hominini-specific deletion in the enhancer region of the CBLN2 gene resulted in its expanded expression across all cortical layers in humans. What was the direct molecular consequence of this specific DNA deletion? A, it created a novel binding site for the SOX4 activator. B, it removed the binding motif for the SOX5 repressor. C, it caused hypermethylation of the CBLN2 promoter. Or D, it converted the enhancer into a dominant silencer.
Think about the severed brake line.
And finally, question 5. During early postnatal human brain development, which of the following epigenetic modifications specifically accumulates in mature postmitotic neurons, contributing to the experience-dependent downregulation of gene networks? A, CpG methylation, B, non-CpG methylation, or MCH, C, 5-hydroxymethylcytosine, or D, histone acetylation.
This one highlights the difference between glia and neurons postnatally.
Take your time with those and definitely revisit the text to solidify those mental models. The mechanisms are complex, but understanding them is the only way to truly decode the architecture of the mind. Keep reading, keep questioning the models, and we will see you on the next deep dive.
Here are the details of this episode. PSYCH 8002. Functional neuroanatomy byline. The architecture of thought mapping structure to function in the living brain.